All right, Jason Curry, week 4, bio 225. This is exercise 1 and 2 introduction. Um, so the exercise 1 is the methyl red and Vogus press core testing. Nothing on the card here. So basically what I did was I grabbed my MRVP broth and I brought that to room temperature. I think it was about 30 minutes. Then while that was getting to room temperature, I got my area prepped, sterilized, ready to go. I labeled my MRVP tubes, E. coli, and the epididymis. Got those labeled, ready to go. And I took some pipettes and I stirred those in alcohol also. And then from that point, I took my the E. coli and epididymis cultures that were done in previous lab assignments, and I got those prepared. Opened them up, sterilized the tip and the lid, and I took my sterilized pipette and I placed it in the coli, and I drew up about 4 drops, and then I placed those 4 drops in the MRVP culture. And then I did the same thing for the epididymis culture also. And then that was all set, and I set it into my incubation area for 48 hours. So after 48 hours, I noticed there wasn't too much growth. The assignment said wait another 24 hours if needed, so I did. I waited almost 72 hours, and I noticed there was a little bit more substantial growth in the MRVP tubes. So I got that all ready to go, cleaned my area again, and I took 4 test tubes and sterilized that into with alcohol and let dry, get ready for the procedure. So I labeled the test tubes E. coli and epididymis, and I placed the MRVP broth equally into the 4 tubes. From that point, I took 2 tubes, the E. coli and the epididymis, and I added 7 drops of methyl red reagent into the E. coli and epididymis tubes. And then with the other 2 tubes, I took the extra E. coli and the epididymis tubes, and I swirled 12 drops of Barrett's A reagent and swirled that into the tubes. And then I took 4 drops of Barrett's B reagent, and I shook the tubes so it would be ready for, it basically got exposed to oxygen. I let that sit for 30 minutes, observed the observations, and I cleaned up the area. Exercise 2, the catalase testing. I gathered my the supplies. I sent the slides, alcohol, peroxide, and the culture plates from the previous labs. I prepped the area again, made sure it was all sterilized, and then I got my the inoculation loops, and I sterilized those in alcohol. And then I got my slides. I labeled the slides E. coli and epididymis, and then I put a dime-sized circle behind the slide, flipped it over, and then I took my cultures and I smeared them onto the area. It basically transferred to the slide. And then I took 1 drop of the peroxide, and I introduced it to the slide with the cultures and waited for a reaction. I cleaned up my area and was ready for the results.